Hi, welcome to biology classes with Lakshmi Chandra. As you know, we are dealing with a series of sessions related to translation in prokaryotes. Already the overall view of what is this translation was given in the previous video, where I had tried to connect the whole process with a cinema. Okay, like in a cinema we have hero, heroine and other cast members. Okay, same way here also I'm just trying to connect it with a cinema. Okay, like here mRNA is taken as the heroine, tRNA as the hero, and they have started a factory. Factory is named as ribosome, and inside this factory they are synthesizing proteins. So actually the heroine is controlling the whole unit. She is asking the hero to bring the raw materials as and when needed. The hero goes and gets it, and then the whole thing is assembled inside this factory. And others which I have mentioned here as enzymes, the initiation factors, the elongation factors and the release factors, they are all the co-workers here helping them to build their factory. So actually in translation it is protein synthesis. So that's what we are going to deal with today. And the whole process of protein synthesis can be divided into 4 heads. Among these 4 heads, today we will focus on the formation of pre-initiation complex, or what is pre-initiation, and also it is also called as charging of tRNA. So we will focus on the first thing, pre-initiation or charging of tRNA. So as it is told here, it is related to the hero, that is tRNA. So the structure of tRNA was dealt in detail with in the previous video. The link for that will be given in the description box. So please go through that also regarding the overall view about translation in prokaryotes. So here charging of tRNA, it is like charging of tRNA means tRNA is linked to an amino acid, any amino acid. So charging means this, tRNA is linked to an amino acid, and for this charging to happen it needs energy. So ATP is required. So ultimately what is produced is you will have a tRNA, and this tRNA will be linked with the amino acid. So you call this whole process as aminoacylation of tRNA. That means tRNA is added to an amino acid. Aminoacylation of tRNA. But as it is shown here as one step, it is not so. It is actually happening in 2 steps. So let me bring clarity on that. What are those 2 steps? Okay. This whole process can be divided into 2, or 2 process happens here. So I am putting it as 1A. That is the first process. In the first process, what happens is the tRNA has to be charged. That means it has to get energy. Okay, and then it is attaching itself to the amino acid. So, or we can say this amino acid has to get energy and it has to go and attach to tRNA. Anyway, the whole process of pre-initiation or charging, it requires ATP. So let us see one by one. Okay. So first process I can say like adenylation of amino acid. Adenylation of amino acid. That is, to amino acid one adenine group is added. From where it is getting this adenine group? It is getting the adenine group from ATP, adenosine triphosphate it is. So amino acid is going to get an adenine group first. Okay, so I can just write here like this, amino acid is added to one ATP. So my amino acid is now adenylated, okay, and then 2 inorganic phosphates are released. So ATP, 1 phosphate is added, or 1 phosphate is taken for this conversion for energy, triphosphate, rest 2 phosphates are coming out here. So my amino acid is now adenylated, or adenylation of amino acid has occurred. And who is helping this? For this, the enzyme aminoacyl tRNA synthetase is helping for this whole process. Aminoacyl tRNA synthetase. Aminoacyl tRNA synthetase is the enzyme that is helping for the whole process. It is having a role here as well as in the second one. Okay. So first step, adenylation of amino acid is done. Now this adenylated amino acid having that adenyl group, it will now join to the tRNA. It will now join to the tRNA. So first amino acid got attached to this one. Now the whole thing is now getting attached to tRNA. Okay. So what will you get now? You will get a tRNA which is having, tRNA is attached to adenylated amino acid. Okay, tRNA is attached to this adenylated amino acid. So you can say what tRNA has undergone? tRNA has undergone aminoacylation. It has got aminoacylation. So these are the two things. So ultimately how our amino acid or our tRNA will look like, let me just write over here. Let me just show here. So our hero will ultimately look like this. This is the tRNA molecule. Sorry. This is the tRNA molecule. Okay, and it is having a 5' end here. 3' arm is a bit longer than 5' end. So to this is our tRNA. To this tRNA who is coming, aminoacylated or amino acid, adenylated amino acid is coming. So adenylated amino acid is coming here and it is getting attached to this tRNA. And as I've told you in the previous session, it is getting attached to the OH group of the 3' end. And so my amino acid is now sitting over tRNA. Okay, so ultimately I can write like this: over the 3' end, over the 3' end, here amino acid is placed. So this whole thing is, you can say this tRNA is attached to amino acid. So I can say aminoacylation of tRNA has occurred, or aminoacylated tRNA. This is aminoacylated tRNA. Okay, so this is happening inside the cytoplasm, or you can say if it is prokaryote, it is happening inside the protoplasm, because we are dealing with prokaryotic translation. It is happening in the protoplasm. So which amino acid should come here, or which amino acid should be carried by a particular tRNA? That is very much specific. One tRNA can carry only one amino acid. It is very specific, like lock and key. Okay, we can have only one key for a lock. Same way here, one tRNA can carry only one amino acid. Another tRNA should carry another amino acid. You know, there are 20 naturally occurring amino acids, and for each amino acid there is a specific tRNA present. Okay, so that is the hero. And which tRNA should come with which amino acid and when it should come. All these I told you in the beginning itself. The whole factory is controlled by the heroine. So it is the heroine which is giving those instructions. And in what form they are giving? It is giving in the form of codons, that is triplets of bases. So regarding that also, it was given very clearly in the previous sessions video. The description is given. In the description you can get the link, and please go through that for clarity. So this is our pre-initiation. So once again I say pre-initiation or charging of tRNA, it is a very simple process. Here the tRNA molecule is attached to one amino acid. This is a simple thing. Okay, and this simple thing happens in 2 steps. First, the amino acid is getting activated by adding it with an ATP molecule, and the amino acid will get the adenine from that. So it becomes adenylated amino acid. Now this adenylated amino acid now can very well occupy its space over the tRNA's OH group. Okay, so now tRNA is carrying the amino acid. You call the tRNA as aminoacylated tRNA or charged tRNA. So there ends the pre-initiation process, and for this whole process the key player or the key enzyme, that is aminoacyl tRNA synthetase. So I hope you have understood today's pre-initiation process. In the next session I will definitely come with the process of initiation of translation in prokaryotes. Wait till then. Thank you all.